Hello, everyone. Thanks for attending my talk today. The title of my talk will be Bile Acids and CYP3A4, otherwise known as cytochrome P450 3A4, drug-drug interaction, or DDI, and FRGN19, liver-humanized mice. For those that aren't familiar for this particular area of research, this is actually a published manuscript. So if you have the opportunity, please visit the Journal of Drug Metabolism and Disposition. This was published originally last year with a variety of individuals stemming from different industries as well as academia, and their names are listed here on this slide if you're interested. So jumping into the presentation, I wanted to start by giving some background for those that aren't familiar with FRG liver humanized mice or humanized mice in general. So humanized mice are just genetically modified rodent models or mice in particular, where the liver is actually ablated. So the murine hepatocytes are selectively removed and then thereby replaced by primary human liver cells, otherwise known as hepatocytes. So in this figure, you can actually see a summary of this particular liver humanized mouse model, FRG. And then the F in the bottom left-hand corner, if you see FAH knockout, that is actually a acetoacetate hydrolase knockout. And so what that does is it actually selectively causes toxicity in the mouse hepatocytes themselves. And then in addition, we also incorporate a RAG2 and an IL2RG knockout, which also causes immunodeficiency that prevents xenograft rejection. So what we're essentially doing is actually knocking out the murine hepatocytes as well as creating an immunocompromised mouse model, thereby giving us the ability to actually incorporate and introduce human hepatocytes to the ablated murine liver. And so what we were actually able to do in this knockout mouse model is actually inject through the spleen these primary human hepatocytes and then apply selective pressure to favor human hepatocyte expansion. So essentially what you get is a humanized FRG or liver humanized mouse model. These mouse models are also referred to as chimeric liver mouse models, given that they're not 100% human hepatocytes. There still are some remaining murine hepatocytes available. So if you can see from the histology of this humanized liver stained with the anti-FAH, you can actually see the majority of the brown stain is the FAH positive human hepatocytes. And then in blue, we have our FAH deficient. So these are the murine hepatocytes that are left over. So essentially, we can get somewhere around greater than 90% in some cases, human hepatocyte replacement index. And so this actually allows us to perform a variety of applications, in particular for drug discovery, to understand human specific drug metabolism without actually going into human studies. So we're actually able to use these rodents to understand human specific drug metabolism. And then using our in vitro and in vivo correlation and extrapolating to humans, this gives us a much better understanding as to what to expect in terms of the drug metabolism that could occur in humans. So moving forward, I just wanted to show that, as I mentioned, there's a variety of applications for this particular mouse model. So I think, in particular, given that they have human hepatocytes, we can actually perform expression profiling as well as apply particular study designs using this mouse model. And in particular, I actually like to use these mice for drug discovery studies, in particular drug metabolism. And so, as you can see from the figure on the left, by introducing human hepatocytes, we can get human drug metabolizing enzyme specific expression, in particular the CYPs, otherwise known as cytochrome P450s. These are major drug metabolizing enzymes in the liver of both rodents as well as humans. And we can also get a humanized expression profile for lipid metabolism, as well as different coagulation factors, complement factors, and in particular, bile acids. And bile acids is very important in terms of normalizing the human expression profile in these mice, given that they actually regulate a wide variety of drug metabolism pathways. And in particular, some of the applications and downstream utilities for this mouse model include drug-drug interaction studies. So if we wanted to understand whether or not a concomitant administration of two different drugs can actually inhibit or induce the metabolism of one another, we can actually use these mice to understand potential drug-drug interactions that could occur in humans. We could also understand human-specific cell therapies and interactions with various metabolic diseases that are human-specific. clinic, as well as other applications such as gene therapy, infectious disease, as well as safety studies and toxicological studies, in particular studying human-specific hepatotoxicity. So these mouse models are also used by safety assessment as well as toxicology personnel to understand toxicity-related indications. So lastly, these FRGN mice were actually initially generated. However, they showed deficiencies. So we saw these very large bile acid increases, in particular that were not human physiologically relevant. And so what we decided to do is take this a step further and then derivatize this mouse model by incorporating another human specific factor, which is human FGF19. And this was able to normalize these deficiencies using a new term, FRGN19 strain, where this FGF or fibroblast growth factor 19 gene was added, and then it helped to suppress and attenuate some of these elevated bile acid levels. And I'll explain that on the next slide as well. So this is just a schematic representation of the physiology or physiological differences between human and mouse specific. On the left, we have human liver as well as human gut. So this is a representative hepatocyte as well as a representative enterocyte, particular to each specific organ. And on the right, we also have our mouse. And one thing that you can actually see very clearly is that the signaling pathways between human and mice actually vary quite differently. So even though we have a humanized liver in this mouse model, the intestine is actually still murine specific. And that actually represents a problem in particular because human enterocytes actually generate FGF19, which is able to interact with FGFR4, thereby inhibiting CYP7A and actually attenuating and normalizing bile acid regulation. So this particular protein, FGF19, is very important in terms of regulating bile acid pathways in human hepatocytes. But in the mouse, in particular, that's actually an ortholog, FGF15, and that's actually not able to bind to the human hepatocytes and thereby regulate these bile acid pools. And so, in doing that and in understanding this metabolic pathway, we decided that by actually incorporating a human specific transgene of FGF19, we would be able to attenuate these elevated bile acid pools. And so this is actually nicely shown on this slide where the FXR FGF19 axis. So this is in particular the pathway that FGF19 actually interacts with is disrupted in the FRG mice. And this is the mice without the FGF19 human transgene, but it's actually restored in the FRGN19 mice. And this was our therapeutic hypothesis in moving into these studies. And so we decided, okay, well, once we actually incorporated the human FGF19, was this actually attenuated? Were these bile acid pools decreased? And were we able to actually normalize some of the expression of these drug metabolizing enzymes? Given that when we have elevated bile acid pools, we can't study drug metabolism or drug-drug interactions in a physiologically relevant scenario, given the bile acids are elevated. And so that causes toxicity. And so that was the overall goal of this project. And so some of the key questions that we presented in this study were of relevance of FGF19 as well as that expression, as well as FRGN mice in particular, were the humanization of the liver in the FRGN as well as the FRGN19 mice. How much was that humanization? Was it successful in terms of the bile acid profiling, as well as the ability to recapitulate a known human physiologically relevant drug-drug interaction? In this case, we used rifampicin as well as triazolam, which is a very well-known induction pathway via cytochrome P450-3A4. And so we were able to recapitulate that. And I'll show some of that data on the following slides. Again, this manuscript has been published as of last year, and a lot of the granularity and details, in particular related to this research, is also published online. And I'd be free to and happy to answer any questions moving forward. So moving on to the next slide, I just wanted to show some of the first data that we actually generated in this study. And as I mentioned, our goal was to attenuate and decrease the total bile acid pools in these mice, given that they were very elevated in the FRGN mice. And by incorporating the FGF19, did that actually normalize and attenuate these elevated bile acid pools? And what we actually saw was this was shown both in the feces as well as in the serum of these mice. So our experimental setup, we had three groups. On the left in red, these mice were receiving a standard diet. These were the original FRGN mice without human FGF19, these still had the murine FGF15. So that disconnect between the gut liver axis was still in play in this mouse model. In green, we had the human FRGN19 mice. So this was the FRGN mice now with the global humanization of FGF19. So now we're able to hopefully attenuate those bile acid pools. And then the last group of mice was the same mice, but we actually gave them a tyrosine-free diet because another secondary endpoint that we were interested in this study was to understand the diet-specific differences in bile acid regulation in these transgenic mice population. And so, as I mentioned, we had an overnight fast, and all we did was collect serum and feces. And then so we profiled a number of bile acids. I think over 50 bile acids were profiled in this study, and all the data is actually included in the supplemental material. But in particular, I wanted to highlight these two figures on the right. On the left in the feces, you can actually see both in the standard diet as well as the tyrosine-free diet and the FRGN19 mice. Both mice exhibited statistically significant decreases in the total bile acids. And in the serum, we actually see a similar decrease, both in the FRGN19 mice compared to the FRGN mice. And so we also revealed that the diet actually has no relevant effect. So that was also an interesting finding from the study. And as I mentioned in the manuscript, there's additional data. In particular, we also saw hepatic mRNA and gene expression reduction of cytochrome P450 7A1, as well as murine CYP7A1 mRNA expression from the residual hepatocytes remaining. And those were both lower in the FRGN19 mice, also consistent with our hypothesis that FGF19 is regulating the human hepatocytes in a fashion that we would expect. So moving on to the next slide, as I mentioned, this was a very data-rich manuscript. So for those that are interested, all the supplemental data is available in the manuscript and published online. But in particular, the bile acid profiling, we actually saw an increased contribution of trihydroxy bile acids in these FRGN19 mice. And this was somewhat surprising. The meaning behind this, we are still unclear. But as you can see on the two left-hand panels, the top and the bottom, the human FRGN feces as well as the human FRGN serum had a large population of dihydroxy bile acids, whereby when we incorporated the human FGF19 protein, we actually saw a reduction in dihydroxy bile acids and an increase in trihydroxy bile acids. And again, the meaning behind this is actually still unclear, but what we've actually noticed is that there's a few relevant examples of decreased bile acids that can actually cause toxicity. And so this was a finding that we were very happy with, given that this suggests that the hepatocytes aren't going under any toxicological events related to these increase in bile acid pools, thereby creating a more physiologically relevant and healthy scenario where we can actually study drug-drug interactions and predict human-specific drug metabolism related events. So this is an additional slide in particular. So I showed on a previous slide the total bile acid pools, but in particular, you can actually look at specific bile acid species in this type of metabolomics analysis. And what we also saw is a reduction in deoxycholic acid, or DCA, in the human FRGN19 mice, both in the feces as well as in the serum. And this is shown by total DCA as well as the percent DCA, which is a relative normalization of deoxycholic acid, in particular normalized to the total bile acid pool. And this is actually a very interesting finding. As I mentioned, the strong reduction of this toxic bile acid is actually really relevant, given that this has been a major hurdle for humanized chimeric liver mice models in the past, given that the human hepatocytes are not necessarily the healthiest. And then this could actually introduce experimental artifacts when interpreting drug metabolism or specific drug-drug interactions. In addition, the comparable DCA levels to non-humanized FRGN animals, this was actually another finding. And lastly, I think this confirms that we are seeing a normalization of this gut-liver communication axis, which is what we intended to do when we actually incorporated this human FGF19 globally. And this is the sort of another interesting finding and last interesting finding related to the bile acid profiling studies that were performed. We saw a reduction in uracolic acid. So for those in the audience that are familiar with uracolic acid, this is typically understood as a murine or a mouse-specific bile acid, as well as lithocolic acid in the human FRGN19 mice. So given that we're actually seeing a reduction in the uracolic acid levels, this suggests a higher degree of humanization relative to the previous iteration of this humanized liver mouse model. So we're very happy to see this, and is actually it goes along with our hypothesis that we are achieving an improved mouse model, in particular related to recapitulating relevant human physiology. And so this led us to performing, as I mentioned, a drug-drug interaction study. So one of the primary tools or primary applications we like to use these mice models in particular in drug metabolism and pharmacokinetics, is to understand the impact on drug-drug interactions, in particular in humans. In this case, we actually used a previously well understood drug-drug interaction phenomenon where by rifampicin actually induces cytochrome P450-3A4, which is the major human drug metabolizing enzyme, both in liver as well as intestine in human. And so what we did is we actually took two groups of mice, the human FRGN19 mice, as well as the previous iteration of this mouse model, the human FRGN mice. And so what we did was administered rifampicin orally, 50 mg per kg, or vehicle, 5 milliliters per kg PO once every day for 5 days. And so what that will do is rifampicin will actually induce cytochrome P450-3A4 by binding to the upstream FXR receptor. And so what this will do is actually increase expression of CYP3A4, which is actually what we want to see, given that we know that this actually occurs in humans. And then after 5 days of administering rifampicin, we administered triazolam at 3 mg per kg, and then performed a pharmacokinetic study to look at not only the triazolam exposure, but also the two primary metabolites of triazolam, 1-hydroxy and 4-hydroxy metabolism. And so we did perform serial blood sampling, and what we can see actually from the two figures on the bottom, we also were able to look at the mRNA expression. And we showed that human CYP3A4 in both groups of mice actually increased in terms of its gene expression after rifampicin treatment. And in addition, the murine CYP3A11, and so this is the CYP3A4 ortholog and the remaining murine hepatocytes, we also saw an increase in expression, approximately threefold in the murine hepatocytes. So this induced human hepatocellularity by 3 weeks, NTBC deprivation. So this is, in particular, NTBC is a drug that we actually administer throughout the development of this mouse model, given that they're very sick. We want to make sure that they actually have complete murine hepatocyte ablation. And lastly, in the figure in the top right, we can actually measure human-specific albumin. And so this gives us an indication in terms of how much humanization we're actually achieving, given that mouse hepatocytes will make murine albumin, and then human hepatocytes will make it human albumin. And we can actually see that there is greater than 70% human albumin in the circulating serum of these mice, suggesting that the replacement index of human hepatocytes is relatively high. And given in together in total, we see successful induction of CYP3A4. We see primarily human albumin circulating in the serum of these mice. We are able to recapitulate this particular human-specific drug-drug interaction in these mice. And so we are very happy to see that. And then on the next slide, what I'd like to share is sort of the final pharmacokinetic data for this drug-drug interaction. So what you can see actually in panel A, so this is the pharmacokinetic profile after triazolam treatment. So again, the rifampicin was administered for 5 days orally, and so that induces cytochrome P450-3A4. And so what we would expect is actually increased metabolism of triazolam. And so what you can see in red, this is actually our previous mouse model, human FRGN. And then in blue, we have our rifampicin treated corresponding mice. And then in green, we have our FRGN19 vehicle treated mice. And in black, we have our human FRGN rifampicin treated mice. And in both cases, actually, both for the human FRGN as well as the human FRGN19 mice, we were able to see a statistically significant reduction in triazolam exposure, which is expected relative to vehicle, which is expected given that with increased cytochrome P450-3A4, you will have decreased triazolam relative to vehicle. And so we are very excited to recapitulate this finding, given that this is a known human drug interaction, and we were able to actually recreate this event in mice. And so this was very exciting and potentially suggests further utility of these mouse models moving forward, in particular for drug-drug interactions that are not known. So screening for potential prospective drug-drug interactions in the case where a particular new molecular entity may cause a risk or may interact with particular enzymes such as cytochrome P450-3A4. And this would be very useful given that we would like to know this as early as possible, especially before pursuing these drugs in clinical trials. As I mentioned, some additional information, which is also available in the manuscript, we saw a significant 3 to 4-fold reduction in triazolam Cmax, as well as area under the curve. In particular, we saw a slight but non-significant increase of the metabolite, and so that actually agrees with our hypothesis in that when you have increased metabolism of triazolam, you will have decreased triazolam exposure, but you will actually have increased triazolam metabolite exposure. And this is actually suggested by the non-significant increase. Though non-significant, we did see an increase in this particular metabolite. What was very interesting and actually very surprising to us was that we actually saw a reduction of the secondary CYP3A4 metabolite for hydroxytriazolam. And we're actually still a little, perplexed by this finding. There were some suggestions by others in the group that I think hold some merit in that there could be some other interactions at play in these chimeric liver-humanized mouse models where we could see further downstream metabolism of 4-hydroxytriazolam in these rifampicin-treated mice. However, further studies need to be performed in order to confirm that hypothesis. In particular, though, the largely comparable effects were seen for CYP3A4 DDI interactions in both mouse strains, and we were able to have good qualitative, however, not very quantitative DDI predictivity for humans. So our suggestion moving forward and using these mice in drug discovery and drug metabolism efforts are primarily centered around using these as a qualitative tool to investigate whether or not there may be a potential drug-drug interaction, as in particular the specific absolute quantitative translation of these drug-drug interactions in terms of fold change of both the parent as well as metabolite need to be further investigated, and in particular, they could still warrant human-specific drug-drug interaction studies moving forward. And that brings me to the final conclusions of this presentation. The first being that physiological FGF19, so fibroblast growth factor 19 expression, so this is the human-specific FGF19, actually normalized the bile acid content in these FRGN mice. And this was based on a restored gut-liver axis shown by gene expression data, bile acid metabolomic profiling data, as well as downstream pharmacokinetic data. In particular, toxic bile acids such as deoxycholic acid or DCA comprise a smaller portion of the bile acid pool in the FRGN19 mice, which suggests that they could potentially be utilized for safety or toxicological studies when looking at human-specific liver tox studies. In addition, the rifampicin-induced drug-drug interaction, or DDI, is not different between the FRGN as well as the FRGN19 mice. So our original hypothesis that by attenuating the bile acid pools in the FRGN19 mice, we were able to increase the event of this DDI, given that bile acids pools can interfere with measuring drug-drug interactions, in particular related to FXR-mediated CYP3A4 interactions. However, we saw from the outcome of the study that actually the activity and the drug-drug interaction findings that were found were actually relatively consistent between both populations of mouse strains. Lastly, FRGN19 mice showed normalized bile acid pools, as shown by the metabolomic data that was presented, and these less toxic bile acids. In particular, we believe that these FRGN19 mice are suitable as models for drug-drug interaction studies based off of these PXR and FXR-mediated induction of CYP3A4 expression. And with that, I'd like to finish with my acknowledgement slides. There were a number of people that were involved in these studies from a number of different industries, from AbbVie, from BMS, Genentech, Ecuris, AdmiScope, just to name a few, as well as Nico Shear, who was heavily involved in consulting for us in this consortium. And I think this has been a very nice presentation, a very nice example of how industries as well as academia could collaborate together and put their heads together to try to push the limits and try to redesign older iterations of both cell models as well as in vivo models for prospective applications in drug metabolism. And I'd be happy to take any questions. I believe my email will be shared. Feel free to reach out. And if not, you can probably find your answer in the manuscripts it was published of last year. Thank you very much.